to our last uh, chapter for our microbiology. So the very specific term for our food microbiology, very important topic for you as food technologists, your food microbiology. So here, the content here is the coverage for microbiology the organism, yeah, and also ilang agriculture sa food, yung mga foodborne illnesses, mga pathogens, mga although discuss na but at least daw i-review. You have your mga food spoilage that is because your microorganism, food fermentations, your microbial physiology, and also food preservation, your microbiological analysis, and for food safety management system. But you have your different types of your microbes. You have your bacteria, fungi, parasites, and also your you have your microbial growth. So have many parasites have complex life cycles. From your bacteria, ang ilang binary fission, ang imong parasites that have different life cycles, ang mga viruses, need nila human host to survive, ang mga mycology, ang mga fungi, okay? So they have different life cycles, the discussion of which are beyond the scope of our purpose here. The most important parasites have your mga Trichinella speculacis, okay, sa tunog pork, okay, cannot reproduce in meat or poultry products. Ang ilang replication sa atong virus will also not be discussed because these microorganisms can only be replicated in living animals. Okay, favorable environmental condition exists, bacterial growth occurs for our purposes. We will use the term growth to refer the increase of your microbes or atong na microorganism numbers in not an increase in the size sa atong na organism. Bacteria produces by dividing, and this is what you call your binary fission. What? Then your bacteria cell is ready to be divided. The material within is gradually increased and the cell's volume is almost doubled. Mga cocci shape becomes oval and mga rod shape stretch to nearly twice their length. The cell then constricts in your middle, and the constriction depends upon until the cell contents are held into two distinct compartments and then i-divide sila sa ilang wall. Two components finally separate to form new cells or ang ilang daughter cells which are duplicates of your former cell or atom yung tawag ng parent cell. Your bacterial cell bacterial group chart, and hindi mo pagkalimutan, you have your log phase, your log phase, and your stationary candidate phase. Tandaan nyo, una ang A sa order for group, have log phase anay, wherein umpisa pa lang multiply ang mga bacteria, whereas the nabaguhan mo siya sa environment. Okay, then you have your log phase, the increased number of your bacteria. Then you have your stationary phase kung sa diin the growth rate or ilang multiplication rate kung ilang death rate is even or ga equilibrium. Then you have your death phase kung sa diin mas less na ang multiplication sa bacteria due to the lack of nutrients. You have your factors affecting your microbial growth. Yan ang balik ko naman sa atin yan, no? I-summarize tayo. You have your food composition, okay, acidity which is refers to the pH. You have your temperature, ang time kung ano sila kaduga ita. You have your oxygen level or carbon dioxide level and your moisture or sa water content sa environment. So for your food composition, here we are using the food for you have here your food composition. Here we are using your word food refers to the nutrients available sa atong mga microorganism which could be found as mga human food product. Your product residue sa atong mga equipment or organic degrees in some non-product contact growth niche. Suitable supply for our nutrients is the most important condition affecting the growth of our bacteria. Every living cell requires a certain nutrients to multiply. This includes the solution sa aton nga agar plates or sa aton mga carbohydrates, proteins, kag sa mga small amounts of other materials such as your phosphate, chlorides, kag calcium. If the nutrient supply is removed, the bacteria will not multiply and the population or sa ilang colony will just gradually decrease. Now, for your acidity naman yung class, okay, it refers to your pH gani. To the degree of your acidity or alkalinity or pagiging alkaline or basic, okay, is an aqueous solution is expressed on a scale from 0 to 14. Refers to as your pH scale. And as acidity increases, we move down on pH scale. Ibig sabihin, the lower your pH, it is the higher your acidity level. As alkalinity increases, move up siya. Ibig sabihin, mas taas yung mga pH value or close to 14, it is alkaline or basic. Then you have your pure of your the neutral level is your 7.0, which refers to neutral pH or sa aton nga water. Because usually itong mga water is in neutral na pH. The pH of the meat or itong poultry product can have a profound effect on the growth of your viability microbial cells. Each species of your microbe goes within an optimal range of your pH value. Most of your microbes thrives when your pH is near neutral or slightly acidic, but there is an exception. Most of the bacteria will not grow on a pH below 4.5 because the environment is too acidic. Many molds or mga yeast can grow in a lower pH than do bacteria. And on the basis of your pH, mga food products are often grouped in a high acid foods, such as mga below 4.6. And a low acid foods, which is around 4.6 and above. The pH or mga fish meat ranges around 5.3 to 6.4 for a high pH or a low acid. Now, for your temperature ng mga class of all bacteria, molds, yeast, and optimum. Okay, you have the word optimum, maximum, and minimum temperature for your growth. The optimum class, okay, the optimum is the preferred and the best na temperature for the bacteria to grow. The maximum is ang ilang limit where they can still grow, and the minimum is the least required temperature for them to grow. These temperatures can vary among different types of mga microorganisms or sa aton nga mga fungi man, and therefore environmental temperature not only impacts the rate of growth sa aton nga microbes but can determine which microbial species thrive or gabuhi. Now, for that time, we have already mentioned the ways in which time can influence your microbial growth, permitting the sufficient time for your microbes to adapt sa ilang environment, which is your lag phase, sa umpisa sa aton nga pag-multiply sa itong bacteria, which is lag phase, is necessary before they can enter a rapid growth, which is your log or log phase. The doubling time for the most bacterial species is between 10 to 30 minutes under optimal condition for growth. Optimal kasi it is the best growing condition para sa imong microorganism. Now, for your oxygen naman yan, okay, similar to your temperature, oxygen availability can determine which microbes will be active. Your microbes can have the absolute requirement for your oxygen, which is your obligate aerobes, and then the requirement for the total absence of your oxygen is what you call your obligate nga anaerobes. For example, obligate, it is 100% ga amugid na siya. Now, some microbes are called your facultative, with the facultative aerobes because they can grow in the presence or absence ng oxygen. So, it is facultative and it's either by oxygen or wala, they will still grow. Now, for your molds, they requires oxygen for growth. For your yeast, grows best under aerobic na condition, but some can grow slowly under anaerobic condition. Anaerobic is wala sa oxygen present. Now, for the moisture, it is the ability of the water in a food, refers to water activity or AW. Now, it is an important factor for the microbial growth. Also, sa ilang nutrient for your microbial growth must take in a soluble form for your microbes to utilize. Generally, bacteria have the highest activity in your water requirements. Mold have the lowest, and the yeast is being intermediate. Ano na ginamit niya para mas lasig mag-multiply o magamit sa imong mga bacteria ang imong mga nutrients sa imong prepared nga media. For microbes ang iyang
food spoilage. Your spoilage is caused by a physical or chemical change in your food product that results in undesirable odor, also flavor, texture, and the change in color. The spoilage costs the food industry millions of dollars each year sa mga na-expired nga mga products. There are three primarily mechanisms that can result in the spoilage of your meat or mga poultry products after your slaughter and during the processing of the storage. One is called your autolyzing enzymatic spoilage, and it is simply due to a normal post-mortem breakdown of your cellular membrane and releases the enzyme that results deterioration sa aton nga product's quality. Another mechanism is what you call your lipid oxidation, which involves your reaction between your oxygen kag fat molecules in the product, resulting in rancidity. And the third mechanism is your microbial spoilage. Okay? A raw food and even certain processed foods are contain your microbes or microorganisms that will eventually cause the spoilage unless they are controlled or destroyed. That's why ano taton nga uh, that's why gina-pasteurize ta aton nga food. Kung sabihin, patay ta ang mga contaminants or mga microorganisms. Your microbes can cause your food spoilage by two basic mechanisms. The most common mechanism is related to the growth of the spoilage microbes and their active metabolism or food components. The other mechanism of your microbial spoilage can occur with the absence of live microbes. As your microbes dies, they release a virus enzymes that reacts with the change sa atong properties ng components leading to spoilage. So aside sa gina-consume sa imong microorganism, ang imong food product, if the microorganism is dead kag wala siya na totally eliminate or nadula gid sa food product, the dead bodies ng imong microorganism will release its virus different types of enzyme which will cause to spoilage. The indication of your microbial food spoilage varies with your microbes or ano types ng microorganism na involves with time course sang spoilage. Bacteria kagang imo nga yeast are typically result in your slime formation and also bad mga rancid flavors and also discoloration such as your gray, brown, or green or mga amoebic nga spoilage bacteria can be important on a vacuum packaged products and can produce distinctive souring due to production of your organic acid kag gases. Ang mga molds, like your fungi, can cause food spoilage, often result in stickiness sa aton nga product surface and usually formation of a creamy black or green colonies with a fuzzy or whiskers-like appearance or ang ilang dough cotton type na itsura sa aton nga mga molds. Sa atong mga microbes kag food preservation, we are generally concerned with the undesirable effect of our microbes on our food, including the spoilage of food-borne illness. However, many microbes are beneficial to preserving certain foods and also controlling some mga pathogenic microbes and producing certain other desirable characteristics in some products. So here are your mga microbes that are helpful sa food production. A primary mechanism through which certain microbes are used in your food preservation in the process of fermentation. Simply put, fermentation results in the conversion of your sugar to simpler compounds by your microbes under anaerobic nga condition. Anaerobic, wala sila na-expose sa oxygen. The fermented microorganism gain energy through the process of your fermentation. The produced substance inducing your organic acid, peroxides, and ilang bacteriocins, they are inhibitory effect on the growth and survival of other microbial species. Therefore, wala sang contaminant nga mag-grow aside sa ga-ferment nga microorganism there. Your microbial fermentation processes are used in the production of variety of your common food, including your beer, mga wine. Okay, some breads, you have also mga chocolate and yogurts. Mga fermented meat and poultry products includes many salamari, summer sausage, mga Lebanon bugulog na, you have certibalat, your choringer, and also your these fermented sausages products typically have the tangy flavor resulting from the process of your fermentations. And also the process of fermentation primarily exerts the preservation effect by increasing the acidity of your product during this process. The pH level of the product is reduced by starter culture activity by appropriate time and temperature factors. The starter culture will generally include your bacteria that produces lactic acid or your mga halin sa lactobacillus species along with certain other bacteria, yeast kag molds are used to impart certain flavor kag color characteristic to your product. The starter culture is being added into your meat and also poultry mixtures and held in your environmental optimum for the growth. Tandaan nyo, optimum gid hindi maximum or minimum because the optimum is the best environment for your bacteria to grow in microbes in your culture. The temperature and also humidity are carefully monitored kag gina-maintain to promote the growth of your bacteria. The starter culture of your microbes are actively reproduced para mag-multiply siya kag magdamo sa imong nga food product. As they do, they compete with other microbes that might result in the spoilage of their product that might be pathologic. The lactic acid produced by your starter culture bacteria, like your lactobacillus, lowers the pH of the product. It is important that the lactic acid is produced quickly because it inhibits your undesirable nga mga bacteria like your toxin-producing Staphylococcus aureus, which is a normal flora of our skin, and the pH is being monitored over time to determine when the process is complete. Generally, the goal is to achieve the pH of 5 or less within a certain time frame. Microbes and food safety. Certain microbes are associated with your food and can also be harmful. We refer to these foodborne mga pathogen. In your meat, mga poultry products, pathogenic bacteria probably account with the largest portion of all our foodborne illnesses. The most important bacteria, sorry. Okay. The most important bacteria pathogens including your Salmonella, Campylobacter, Listeria monocytogenes, or your LM ilang shortcut, your Clostridium butyi, kung sa diin halin niya sa usually sa mga canned foods, your Clostridium perfringens, your Bacilli cereus, Staphylococcus aureus, and the Shigan toxin produced by your Escherichia coli. Of this, your Salmonella and your STX is the two common cause of your large outbreak. Or ang imong hashtag ni is Shigella toxins produced from your E. coli or hadin sa E. coli. Now, your molds and your yeast or ang imong mga fungi do not appear to be responsible for any meat or poultry-related foodborne illnesses. Maybe because the significant molds or yeast growth is easily recognized as the food and the food discarded. Some molds do produce toxins like your just produced by Aspergillus species, but foodborne related mga illness appear to be associated with your nuts, grains, but not meat and poultry. Also, some molds on your food product may cause allergic reaction and also respiratory problems in sensitive individuals exposed to your molds. Some important foodborne parasites like your Gardella duodenalis, your Cryptosporidium parvum, your Cyclospora cayantinensis, your Toxoplasma gondii, your Trichinella species, mga Taenia, mga tapeworm, and also ang tapeworm sa mga pork which is your Taenia solium. It is important to note that while Toxoplasma gondii is not common cause of your foodborne illness, it is among the top 5 foodborne pathogens that result in hospitalization or death. Your Trichinella or trichinosis caused by your Trichinella spiralis, obstetrically an important foodborne illness resulting from a consumption of mga undercooked or mga hilaw nga mga baboy. And your trichinosis is largely been eliminated due to changes in your swine production practices. Also, ang consumer education kasi di nga napabalugid nga dapat well-cooked ang mga meat products and prescribed treatment for destruction of trichinella in certain cases of food products. Now, sa aton nga controlling sa aton nga mga microbes in food and food processing environment. So your pathologic microbes are significant threat to the public health. There are two fundamental ways to control your microbial contamination or sa aton nga products and processing environment. Both are very important. The first involves the reducing of opportunities for your microbes to enter the processing environment and come in contact with your product. Ibig sabihin, ang mga workers. The second involves with making the environment for your microbes is inhospitable as much as possible to reduce their number and minimize their growth. Note that the word control is a key term here. It is important to completely eliminate all microbes from processing environments and food products. However, it is possible to establish or establishments to implement effective control strategies designed to protect against your pathogens and undesirable effect of your spoilage organisms. Other portions of this training program will familiarize you with specific processes, including specific methods for controlling your microorganisms in those processes. For now, we will be broadly considering the ways in which levels of your microbes is in processing environments and on foods may be controlled. For your prevention and your contamination, it is important to avoid contamination of your meat and poultry whenever it is possible. This includes inadvertent contamination of cross-contaminated from live animal. Your processing procedures and also mga equipments nyo, okay, ang mga employee, mga workers, and the environment or ang mga workplace. The contamination can be minimized or avoided altogether by following appropriate sanitation procedure, ang good manufacturing procedures or your GMPs, and produces for employee hygienes. Good sanitary dressing, process control measures in your slaughters, process not only minimize the contamination of the carcasses or mga bodies ng dead product, but also reduce the level of processing your environmental contamination. Effective pest control, kasi di ng mga rats, mga cockroaches, mga muna, can help prevent the introductory of many microbes into processing environment. Also, your sound construction of your facility and maintaining it construction will reproduce the opportunity of your microbial contaminating on your processing environment. Now, sa itong restriction or pag-stop sa growth sa itong mga microorganism. Okay, recognize that the bacteria will be present in meat and also in your poultry. It is very important to keep overall number of your bacteria very low to minimize the concern about your bacterial pathogens as well as your spoilage organisms. Making a microbes environment as inhospitable. Ibig sabihin, hindi sila grow sa mga environment can involve in variety of control measures. All of them relate in your fat tom factory in plants, microbial growth, effective procedures for cleaning and sanitizing your facility, provide the foundation for controlling your microorganisms. In addition to this, you have your temperature, acidity, salting, or yung mga Na concentration, kag also drying, and some combination that these can be used to restrict the growth of your pathogens. You have your sanitation, temperature, acidity, salty, and drying, kag vacuum packaging. Ultimately, there are no single method for preventing or controlling your microorganism in your food. It requires so-called multiple hurdles to approach, and also the fundamental. This can be represented by compliance with your sanitation performance standard, maintaining the effectiveness of this sanitation or standard operating procedures at mga SOP, and designing and implementing the effectiveness of your hookup plan. Additional notes. Okay, additional notes lang for mga halin sa CDC, okay, mga past studies sa mga foodborne ng mga pathogens. Hindi tayo pag-discuss dito, magpadalman dito. This is statistical data sang CDC. So let's just move forward sa itong mga keywords. You have here mga keywords, liwat. Important sa itong microbiology since this is the last chapter na kapila ni liwat ang mga terms. If you say aerobic, these are bacteria that requires oxygen to grow and will grow in the presence of oxygen. You have also your anaerobic. This is bacteria that do not utilize oxygen to grow or will not grow in the presence of your oxygen. Then you have bacteriocin. Okay, it is a proteinous substance produced by your specific bacteria that is toxic to closely related strains of some specific bacteria, and it either kills or slows the growth of other bacteria. Then you have your coliform. Okay, bacteria that is most often inhibits your intestine and animals then do not utilize oxygen because usually sa aton nga intestine is anaerobic. And but can grow in its presence. Your bacteria that are classified as your coliform have the same shape, and many of these same characteristic of these bacteria are used in indicator of your sanitary quality in many food products. Then you have your term detection limit. It is the lowest threshold of any bacteria that can be present in a sample or to be found. And the detection level depends upon your analytical method being used. Then you have your direct plating. It is the application of a sample, also ang dilution thereof, to the solid media usually containing an agar and other material used to grow and enumerate your bacteria. Then you have your D value. It is the amount of time needed to destroy one log unit of a specific bacterial pathogen at a specific temperature kag specific nga medium. You have enrichment. It is the additional nutrient-rich broth or para sa bacterial growth that will be so that a certain bacteria or type of bacteria increases in number to result in your bacterial cell count that is higher than the detection limit. This is used to detect only the presence of or absence ng mga bacteria, not the amount present. Then you have your no, entero, Enterobacteriaceae, a large group of bacteria that are closely related to commonly found in a fecal material or warm-blooded animals. This includes your coliforms and pathogens such as your Salmonella. Then you have your F value measures in minutes. The D value is specified organism in a 250 degrees Fahrenheit or 121 degrees Celsius multiplied by the desired log reduction. Then you have your germination. It is the process of a spore becoming a vegetative state. Your inhibition, it is the slowing or stopping sa aton nga bacterial growth. Then you have your lag time. It is the time where your bacteria take to become acclimated or sa bago-yang environment maka-adjust, okay, before starting to multiply exponentially. Then you have your lethality, okay. From the word itself, it is the effectiveness of your treatment to destroy or kill your bacteria. Your log unit, okay, it is the log for short for logarithmic. Okay, sa aton nga math, though it is the power or exponential nga multiplication. Pero sa aton pag-apply sa microbiology, it is the multiplication or exponential multiplication of your bacteria. Then you have your mesophiles. Your mesophiles, ibig sabihin na bacteria that have the optimum growing temperature between 77 degrees Fahrenheit or 25 degrees Fahrenheit. That is your room temperature to and your 104 degrees Fahrenheit or 40 degrees Celsius. This usually sa body temperature among mga bacteria which can easily grow sa aton human body. Then you have your microflora. It is the community of microorganisms including your bacteria, molds, yeast that lives in or sa other organism or living organism in a particular habitat. Then you have your pathogens, okay, organisms that cause illness. This organism includes your bacteria, your protozoa, or viruses. Then you have your pH, level or acidity sa aton alkaline in the product. The pH scale range from 1 to 14 and the 7 is considered your neutral and the pH 1 is the most acid and the 14 is the most alkaline. Fresh meat usually has the pH of 5.6. Then you have your cyclorotrope. Okay, bacteria have the optimum growing temperature between 68 degrees Fahrenheit or 20 degrees Celsius and 86 degrees Fahrenheit and 30 degrees Fahrenheit but can grow at a temperature as low as 32 degrees Fahrenheit or 0 degrees Celsius. Then you have your shock or heat shock. Of course, when the product is heated but the temperature is not high enough to destroy the bacteria. This results to a bacteria that are injured for a while but in those cases can repair themselves, okay, and more resistant to heat the next time the product is being heated. Therefore, ang imo heat shock can also refer to the process by which the bacterial spore is induced in a germination. When the product is heated thoroughly, the vegetative state or isang bacteria mo will be destroyed, but the spores are undamaged by the heat. The spore then germinate into vegetative state once the temperature has decreased to a optimum level or mabalik siya sa normal yang stage. Then you have your spores. Pure spores are highly resistant or dormant structure formed in response to adverse environment condition. Bacterial spores are usually very resistant to heat and long period of dryness and the other adverse condition that normal vegetative cells cannot survive. When the environment becomes more favorable, then the spore can reactivate itself to vegetative state. Most of the time, spores-forming bacteria have toxin associated with them. When a reactivation or germination of the spores occur, your metabolic activity begins. The conversion of your germinative spores into a growing vegetative cell, thus producing associated toxin. Then you have your mga strains. Okay, ano ng mga strains? Strains is a genetically variant or mga subtype. Parehas bala sa COVID, you have mga Delta variant. So these are different strains of your COVID. For example, your E. coli here. So you have 015787 strain of your Escherichia coli 57 in a genus coli, and the species is 015787 is a serotype, and the strain EC57 is the strain. Then assign the number of identifier for each genotype. For example, you have your genotypic nomenclature involving specific gene variation or mga heat resistance. Then you have your okay, thermotolerant bacteria that can withstand higher than normal temperatures. You have your toxins. You have your enterotoxin, mycotoxin, neurotoxin are compound produced by a bacterium or your fungi. Your fungi is under sa has your molds and yeast category and can cause illness on other living organism. Your specific example, including your enterotoxins, affects the intestine. Your mycotoxin, those products, your toxin produced by your fungi, and your neurotoxins attack nervous system. Then you have your transdermal synergies, okay, compounds that work with other compounds against bacteria when applied to a surface or a carcass. Ang treatment ng class, okay, the treatment is the method of processing that being tested. A good research study will compare various treatments such as the level of salt in your product to control mga muna sa itong mga bacteria. In this example, the control may be no salt added and all other conditions should remain the same for the sample tested except for a specific treatment. Vegetative state sa itong mga bacteria, it is the normal bacterial cell that is in contrast to a spore. Your vegetative cells are susceptible to destruction or damage from your heat. Mga additives from other factors can damage and destroy them relatively easily. So that concludes aton nga chapter 8 sa aton nga microbiology. So God bless, good always class, and stay safe.